Well, good morning, good afternoon, and good evening to everyone, and welcome to this IWA webinar organized by the Microbial Ecology in Water Engineering Specialist Group. My name is Jorge Rodriguez. I'll be the host of the webinar today. I am based in Khalifa University in the Emirates. So I would like to, again, welcome you. Thank you for your interest in this webinar. We have titled the webinar, Innovative Modeling Approaches for Microbial Ecosystem Engineering. We put this webinar from the specialist group thinking on looking at the modeling of these microbial systems that we all care so much about under different perspectives, let's say outside what is the beaten track conventional biokinetic models. Before we start, we have some housekeeping information from IWA, just to remind you a couple of things. The webinar is being recorded, and that will be made available later on the IWA Connect Plus. So if you miss something, you can always go back and go through that. The speakers are themselves responsible for the copyright permissions and regarding the work they are presenting. And again, just a reminder that the opinions and all the information is the responsibility of the speakers, and IWA is indicating us that it is not reflecting the opinion of IWA. In order to interact with the webinar, there are two options that you might see. There is a chat window, a chat box option that is used only for general requests and interactive activities. Please put all your Q&A questions, the questions for the speakers, in the Q&A box, because that's where I will look into to select the questions. So please don't put them in the chat, because I will not see them most likely. I think that's all the housekeeping we have. I would like to give the, well, this is going to be the agenda today. I'll talk about it in a minute. But before that, I'd like to give the floor to David Weisbrot. David is the secretary of the specialist group, and he has some words about the specialist group before we start with the webinar. So, David.
Thank you, George. I'm happy to speak here from NTNU in Norway in the snow, as behind me. We have a bit more light now. And so, yeah, welcome from the MIWI Specialist Group. And I'm also representing here my colleague Cindy Smith from the University of Glasgow, who is the chair of the IWA MIWI Specialist Group, where we focus on microbial ecology and water engineering. Next. So the aim of the specialist group is to advance the engineering of microbial systems in the water cycle using emerging technologies and concepts in microbial ecology. And there, mathematical modeling plays a key role to better understand and predict what happens in these microbial systems. So MIWI aims for research, dissemination, and collaboration with practitioners worldwide. So we have a strong link with the water industry through IWA, and we want to address critical water sector challenges through microbial systems analysis and engineering. So the current focus of the group is to understand microbial diversity role in water systems and how we can drive collaborations with the industry to develop innovative solutions based on the microbial ecology concepts. So we support the link between IWA and the International Society of Microbial Ecology, ISME, and we collaborate with other specialist groups like the Biofilm Specialist Group or the Environmental Engineering Education Specialist Group and nutrient removal and recovery, as well as different clusters like the wastewater-based epidemiology surveillance cluster. We have 3 awards that we deliver every 2 years at the international conference, namely the Early Career Award within the 5 years after the PhD, the Mid-Career Award within 15 years after PhD, and the Arden and Lockett Award for more established scientists. We represent more than 570 members from all across the world, and we are very happy to have you on board today. And I have a final slide to show the next conference will be at Georgia Tech in Atlanta in USA. The dates are the June 2nd to 5th, of this year, so June 2nd to 5th in Atlanta, USA. It's the 11th Microbial Ecology and Water Engineering Conference, where we want to look at how we can manage water microbiomes. This is organized by Professor Amit Pinto from Georgia Tech and his colleagues in the USA. So have fun with modeling about microbial systems today, and thank you so much for your presence.
Thank you, David. Thanks for the explanations about the specialist group. So those interested, of course, maybe consider joining the specialist group. That would be great to have more people here. So this is the agenda for today. We have modified slightly the order. We have four really excellent speakers with four different approaches. We hope you will enjoy this. I'm going to just not, without further ado, I'm going to just introduce the first speaker of the day, which is Rebecca González Caballero. Rebecca is going to present, the title is Quantification of Microbial Ecology through Bioenergetics Analysis of Metabolic Activity. Rebecca is based in TU Delft. Currently, she's assistant professor in Delft University. She did her PhD in the University of Santiago de Compostela. She has been working on modeling of fermentation systems for many years. I hope you will enjoy her presentation. So, Rebecca, whenever you're ready.
Thank you, Jorge. Let me control the screen. So, hello, and thank you so much for this nice introduction. As you see, my title is quite long and involves maybe difficult words like metabolic pathways and bioenergetics. To understand microbial ecology or to control microbial ecology, that is what brings us all together here, right? You will see how the presentations of my colleagues, they tackle different aspects of modeling, and they will also present completely different concepts. So I thought that maybe starting this meeting with asking us what is a model and how different models could represent the same reality. So as I was saying, what is a model? Because here, and this is a slide that I always present to my students in the master, in the class of modeling for bioprocesses. I ask them, which of these two models of the reality is the correct model? And of course, the answer is that none of them is a correct model, but both of them represent the same reality with different information. So the first model, Las Meninas from Velázquez, has the information that the most important figure is the monarchy, but maybe Picasso, that came later, put more emphasis on the fact that on the figure of the painter, because he had great admiration for this painter Velázquez. That's why we see this big figure on the left, right? So in reality, we are seeing the same reality, but the information that we receive from the model is completely different. So what makes a model a good model? Well, one way of answering this question could be saying that using the sentence that we probably already heard several times, every model is not perfect, but some of them, they can be useful. And this is what we will try, or I try, with the models that I develop in my professional practice. So what I'm trying to do always is to generate a model that can help us with experimentation. And this is what this presentation of today try to put the focus on the integration between this experimental approach, or how we can improve experimental approaches to generate models that are useful to develop new technologies. So in order to do this, we need to consider all the cycle of creating a model. Creating a model is not only generating the mathematical equations, although it's a big important part of it, but also verifying that we implement these mathematical equations in a correct computer model and without mistakes, of course, and then that this computer model can actually be validated. In order to have this complete cycle from creation of a model, verification, and validation, we cannot look only to the model, but also to the experiment, because we have to be sure that what we can measure in the real life will tell us first if the model is correct, and then if the model is useful, if we can use the information that we retrieve from the model to actually change or improve our reality or what we are trying to do with it. So in my specific case, what I'm trying to focus, what I try to do with my models is to use them to help me to select microbial communities, to engineer microbial communities. So in our lab, what we do is we take an inoculum that it can be as simple as any environmental sample. Sometimes we go to the pond next to the faculty, we collect some water, and with it, we inoculate a reactor, but the reactor will have very defined and controlled conditions. These will be the selective pressures that will make this microbial community evolve and eventually stabilize into something that might be of interest for us. But the question is, how do we model this that it sounds quite complex? As Velázquez tried to do with his painting, in which he was trying to capture the reality, we have to capture a reality that is far too complex. We cannot model everything. So we have to figure out what are the the things that we want to look at in order to create a model that is useful. And to do this, the first thing that we have to be sure is that the data that we obtain from the experiments that we run are reliable. And this is a big, massive thing when we talk about enrichments and complex communities of microorganisms. And even when we experiment with pure cultures, we are seeing that the possibility of having parallel bioreactors, as we have here at TU Delft, help us enormously. First, because it allows us to ensure or to be almost sure that each enrichment that we do in these parallel bioreactors faces exactly the same conditions. And then we can maybe decide of one variable that we want to change, and then compare how the microbial community evolves or reacts to this only parameter or variable that we are changing. Sometimes when we read the literature, comparability between experiences is difficult, it's too complex. Or when we try to look for a kinetic parameter, we find that the range of orders of magnitude for that parameter is too huge to really extract some conclusions or some data that can help us to model. The second important thing that this kind of setup gives us is a higher resolution. And what I call higher resolution is the fact that we, at the moment, can obtain online data from the gas composition of the gas phase that is produced in these bioreactors. For the liquid composition, we don't have it online yet. We are working towards it, but we also can totally characterize the liquid composition and close our mass balances, carbon balances, and electron balances. But you will see, I bring you here an example. Here, an example in which we clearly see how to have this online gas composition is quite crucial in many cases. In this case, what you are seeing is the result of a fermentation in fed batch in which we do an enrichment with ethanol, where we feed ethanol and we enrich the we grow an enrichment already. The enrichment was done outside of the reactor. We grow an enrichment of 3 populations. If we were not having the online data, that is the red curve, you see that the liquid composition is not telling us what the online data or the gas phase is clearly showing us. When we look at the online measure data, we clearly see that there are 2 curves very well defined, showing that the fermentation occurs in 2 steps. And the first step runs until ethanol is depleted. The second step is a fermentation with the rest of acetic acid, and it occurs in a slower process. From these curves, we already can obtain kinetic parameters and quite precise. And this is very relevant because it allows us to extract some conclusions. When we have a lot of data, we can see that we can extract conclusions like this one. So as we see, when we have a big amount of data and this data has an elevated precision or high precision, we can obtain conclusions like this one. When we observe how microorganisms, and this is in an aerobic environment, oxidize a certain carbon source, we always observe that there is a limit for the respiration rate. And this limit is around of a predicted limit already that it was predicted already in the 80s, around 3 electrons transferred per mole of biomass per hour. So the precision of the data that we can obtain will give us some information that we later can use to establish general understanding of how microorganisms are limited in their activity and how then the selection happens between different populations. So we can think of doing things even easier on the experimental side of things to be able to quantify better. So I showed you an example in which we were having 3 populations competing between them for the substrates available. But we also wanted to make things even easier, having only one microorganism, but this microorganism will have to select between the different metabolic pathways available. Some of the microorganisms, or a lot of the microorganisms that we see in the environment, they do it. They have this metabolic flexibility. We wonder if this metabolic flexibility reacts to selective pressures in the same way than when we have a mixed community in which different populations have to compete between them. So this kind of experimental setup will allow us to compare if the pure culture will comparatively act in the same way of a mixed culture with the same genomic information. But again, for doing this, we have to plan our experiment carefully. So this pure culture, Clostridium pasteurianum, we are growing right now at the lab, typically grows with yeast extract. It's an easy and fast way of growing this bacteria, but we are removing it and we are growing it with minimal media. Why? Because like this, we ensure that we can characterize every carbon molecule that enters in the system, and therefore we are able to then predict what goes out. The high quality of the experimental data that we can obtain from this allow us to also generate parameters that clearly show how this microorganism grows with a high precision. And these parameters can be even more detailed than the typical parameters that we obtain in another more simple experimentation. Like this, we can look at how much ATP the microorganism is producing per unit of time at different dilution rates, for example. This allows us to see how the fluxes fluctuate between the anabolism, the catabolism, and partially how the substrate is consumed to maintain the cell. And this we can now quantify it with numbers and also use it to validate models or to decide which parameters we should include in a model that predicts the fluxes. But for example, in this kind of system in which we have different pathways, could you already, just looking at the pathways, predict which will be the most or the preferential pathways of a Clostridium pasteurianum that grows without any limitation? I could imagine that I feed enough glucose, a lot of glucose. Which of the pathways the cell will do first? Well, I think for me at least, the answer will be that the cell will want to oxidize the substrate as much as it can, because it will allow it to increase to obtain the highest growth yield possible. And for this, the cell has to produce a lot of acetic acid. But then we are in a fermentation, so we have to balance the electrons because there is no external acceptor of electrons. So acetic acid cannot be produced by itself. Therefore, the electron balance can be closed if we produce butyric acid, for example, that produces is the only other product that produces 1 ATP when butyric is produced concomitantly. So therefore, I will already predict that most likely, if nothing is limiting, this microorganism will produce a combination of acetic and butyric. And this is what we see at different dilution rates. So in our experiments, what we were trying to do is to force the microorganism by increasing the dilution rate of the reactor. Can the microorganism grow faster and faster and faster from 0.1 of the mu max up to the mu max of this microorganism? What we see is that even if the microorganism has to run faster, it constantly selects the same catabolic pathways. And this is the equimolar production of acetic acid and butyric, that, as we see in the left graph, it produces quite high ATP yield per mole of glucose. So logically, they will produce these two products. The question is, why equimolarly? Well, I didn't bring the calculations, but doing an analysis on driving force, we see that the production of equimolar acetic and butyric releases a high amount of energy. So at the same time, it's producing one of the highest ATP yields per mole of glucose, but also releasing enough energy, allowing the microorganism to run faster when the dilution rate increases. This kind of conclusions is what we are going to use to create new models that can help us to predict relative abundance or relative activities of microorganisms doing different metabolic activities. And this is what we have been doing already for other kind of systems in which we directly were able to compare the activity of microorganisms with what we observe experimentally at the lab. So we have been able to do it already. And that's it. With it, I wanted to mention again that one of the most difficult things to work on when we are doing microbial community engineering is on the integration between the theory and the experiment evolution or advancement to work together to understand better what we see in the lab. Thank you so much.
Thank you very much, Rebecca, for the presentation and for staying in time. We have maybe time for one question. There is a question from the audience, probably. Yeah. So everybody's asking, can the first model be used to measure RNA activity? I'm not sure if this is...
Which model? RNA activity.
Yeah, probably that question. Could you answer that? You answer that?
Well, what we can do is to model metabolic activities, and then from there we could infer some molecular activities or molecular data. We are working towards trying to see if we can predict or compare the metabolic activities that we predict with proteomics data, but I never worked with RNA or transcriptomics, so I will have to look into it.
Maybe I can ask you a little bit of a question. In the new research that you are doing, what are you applying now? I mean, you work before in metabolic and bioenergetic models. You still think they can be useful to tell us regarding this rate yield trade-off a little bit? Do you have any interesting stories about it recently, perhaps?
Yeah, and indeed we still use it, right? This is in the background of what we do, but we are more interested on looking into what is limiting the system or the microbial activity, and this depends on the environmental conditions first, on the environmental conditions that we are studying, and not always is a rate yield trade-off. This rate yield trade-off will also depend on what is limiting in terms of rate. And this is a question that we are asking us right now a lot. It is the capacity of producing protein, what is reducing the rate of a microorganism or limiting the rate of a microorganism, the capacity to transfer electrons between electron carriers, the electron transport chain. We don't know exactly what is limiting the rate of a microorganism, and this might be different depending on the system that we are studying. So it is always in the back of our minds, this rate yield trade-off, but we want to expand it. We think that in many cases it's limited, and in some cases different groups might understand different things by this.
Thank you, Rebecca. That's great. All right. We can move on now to the second speaker. We'll have again, I'm reminding that we'll have a session at the end in which everyone can throw in all your questions, and we'll have a discussion together with the speakers. The next speaker is Georgi Bajic, which is also assistant professor in the Department of Biotechnology in TU Delft. Georgi has a PhD from Spain, from the Autonomous University of Madrid. His PhD is in computational biology, and I think he did also a master's in biophysics and a bachelor in biochemistry. So he has a very nice, perfectly balanced background to deal with this kind of systems we are dealing with here. His research tries to predict a species assembly in community from their genomes, and he's also trying to predict the collective function from the species composition. And the title of his talk is Using Fitness Landscapes to Engineer Microbial Communities. So I think we're looking forward to hear from you, Georgi.
Well, thanks a lot, Jorge, for the nice introduction and for inviting me to be part of this webinar. Let me see first if I can control. Yes, I can. I think I can now. Let's hope it stays. Yeah, so it was really nice to hear Rebecca. I'm gonna be changing topics quite a lot. It's really amazing to see what work people do to characterize mechanistically the physiology of microorganisms, especially microbial communities. This is extremely brave. I'm gonna be taking a shortcut, a statistical one, so I'm not going to be looking at mechanism, and furthermore, I'm going to be looking at a type of communities that are a bit different from what perhaps you guys are used to in this community. And I'm going to be talking about how to engineer synthetic microbial communities. So communities where we are putting together defined species, to try to achieve a function of interest. Right. And the models I'm going to be talking about are based on fitness landscapes, and I'm going to explain what this is for. But to start my talk, I hope there is some basketball fans in the audience. In basketball, it's a team sport, and we have 5 players on the court usually, and another 7 players on the bench. And the coach can make as many changes as they want, right? So when the game is not going well, the coach faces the dilemma of which player to put into the court. And this is typically, approached, based on intuition, right? But based on the individual characteristics of the player. So if I'm scoring, not enough points, then I'm gonna put on the court a player that is good at scoring points. If I need to pick rebounds, I'm gonna put a tall player, and so on. And the problem with this individual based approach, based on individual traits, is that players are interacting, right? And the contribution of a player is going to depend a lot on how that player interacts with the rest of the team. And actually, in engineering, synthetic microbial communities, but also many other fields, we face a similar problem, right? We face a similar problem whenever we have a system where a collective function is delivered by interacting components. And that can be when designing a consortium of microbes to perform a function, for instance, a plant growth promoting function or production of a compound, but also for instance, for designing antibiotic cocktails, because the antibiotics are going to interact with the bacteria they're trying to kill through the physiology of the bacterium, to optimize media for growth of microorganisms or cells, and many other similar problems. So the main question that we face when trying to engineer these systems is what combination of components, which components should I need to add to the system in order to maximize a desired function, provided that these components are interacting, right? And the main tool we use, or that I've been working the past number of years, is the fitness landscape, right? So a very simple explanation, a fitness landscape is basically a map between genotype and fitness. And this has been a very useful tool in evolution, because as you know, in evolution, populations are climbing these fitness landscapes by means of mutations. Mutations are moving these populations through genotype space, and through selection, they are moving towards regions of higher fitness. That's how people conceive evolution and think about evolution in terms of a fitness landscape. And fitness landscapes, again, they are composed of genotypes, but all these genotypes, or these different mutations that can happen, they interact, right? So if you imagine, for instance, glycolysis, well, mutations in different elements of glycolysis are going to depend, of course, on whether glucose is there or not, or other molecules that might be derived through glycolysis. But also, and very importantly, by interactions, right? If a mutation happens in one of the enzymes of glycolysis, that mutation might have a different effect depending of other mutations that are present in the same enzyme or in other enzymes of the pathway, right? So this is just to say that interactions are important in shaping these fitness landscapes. Furthermore, fitness landscapes, are not just an obscure concept in evolutionary biology, but they have actually been useful for engineering. And the example that I always like to show is the work of Frances Arnold, who extensively used fitness landscapes for her work on engineering enzymes. Okay, so how can we use fitness landscapes to engineer microbial communities? Well, you can think of a fitness landscape as actually a much more general structure function map, right? You combine elements in the structure in order to deliver a function, right? And your combinatorial structure, in this case, I'm going to be talking about the composition of a synthetic microbial community. So instead of which mutations, which species are in the community or not. And the function can be whatever you're interested in, the production of ethanol, the degradation of a pollutant, or whatever you are interested in and you can quantify, right? And this is what we call a structure function landscape. So it's a generalization of the fitness landscape. And now it turns out this is a statistical problem because, the number of combinations, the combinatorial space of these fitness landscapes grows as 2 to the n being the number of species that we have. And then experimentally, it is impossible, as this n grows, it's impossible to map all the possibilities, right? So we need predictive tools to try to to try to predict, from a limited set of experiments, to try to predict the shape of this landscape, and then just select whatever composition of our community maximizes the function. Here I'm putting plants. You will see why in a bit, but you can think of microbes as well, and whatever. Okay, so hopefully I've explained a little bit what's the problem, and how we can think in terms of fitness landscapes and structure function landscapes about this problem of trying to come up with the composition that maximizes a given function of a microbial community. I want to introduce now a concept called global epistasis. And this concept is, say, a new way to look at interactions, right? So imagine that I have the following experiment now in fitness landscapes, right? I have different genetic backgrounds, which are here defined by these different colored genes, right? And I have a focal mutation, which is this red gene. And I do the experiment of performing this red mutation in all these different genetic backgrounds. And now I'm going to plot on the x-axis the fitness of the genetic background, which I can measure. And on the y-axis, I plot the fitness effect of adding this mutation, right? And then I start doing that. And for one genetic background, I get, for instance, this value, this effect of mutation I. I do it for another background and I get this effect. I keep doing it. And actually, if this mutation is interacting differently with the different mutations present in each background, what I expect to see is something like this, right? Something quite random. So the effect of this mutation is going to be different in every background. So there's nothing to predict here, right? It's just a cloud of points. But well, it turns out that people have seen in genetics that this often looks like this, right? Not necessarily with this slope, but there's often a linear correlation between the fitness of the genetic background and the fitness effect of mutation I, right? This has been seen in many different organisms, bacteria, in yeast, for many different mutations. It seems to be a widespread phenomenon. And I won't go into the details. We're starting to understand where this comes from and how can we understand this in terms of the classical, pairwise interactions and so on. But the important thing about these relationships from the point of view of engineering is that we can actually use this for prediction, right? If I run a regression and I have just a new, let's say I have a new background, I can predict using this regression what is going to be the effect of this mutation in that background without necessarily measuring it. Okay, so we thought that if we are thinking in structure function landscapes as a more general thing, we asked, is there such a thing as global epistasis also in structure function landscapes of microbial communities? And now I'm going to do the same experiment as before, but just instead of genetic backgrounds, I have ecological backgrounds, so different assemblages of species. And then I'm going to add this red species to each of these ecological backgrounds and measure a given function, right? That function can be whatever I'm interested in. So we did this, actually, we did one experiment, but we tested this in many different data sets that we downloaded, and this is what we saw. So this is a data set with plants, where people measured the function was the plant biomass in different plots where they were introducing. There were communities of plants, right? And they were introducing each of these species. So just to remember, here I'm showing, different, on the x-axis, different plant communities to which I'm introducing this species L. vulgare, and I'm measuring the effect of introducing L. vulgare on the biomass of the plant community. And you see again that we see these global epistasis patterns, these linear correlations, right? We looked at phytoplankton biomass communities. Again, global epistasis patterns. Bacterial xylose oxidation, again, linear correlations between the effect of adding different species and the xylose oxidation that was observed in the background. Starch hydrolysis, this is our experiment. We again observed these patterns. Butyrate secretion communities, again, these patterns, right? We were extremely excited about these results because this could allow us, as I told you, to make predictions. So imagine that I have now, I start with a community of bacteria where I know the function, and I want to add a species, this purple species, and if I know for this purple species the shape of this global epistasis pattern, meaning that if I know the parameter A and the parameter B, which I estimate from experiments, and I know S0 also because I measured it for this community, I can then predict what is going to be the effect of adding this species in my resulting community. And I can keep doing that for the green species, for species K, this blue species, and so on. And I can keep doing this and map the entire landscape, right? And if I map the entire landscape, that means that I'm predicting out of sample community functions. So how do these predictions work? Well, this is an example of how they work. So I'm plotting the predicted function versus the measured function. In this case, it's pyovertin production in an experiment that we did in our lab. Well, in the lab where I was previously a postdoc, this was done, okay? And here you can see the same plots for the different cases that I show you, so plant biomass, phytoplankton biomass, xylose oxidation, and so on, right? So the predictions seem to be working quite well, and we were very excited with these results. We think that this could be, a very interesting method to predict the function of microbial consortia. And do I have time, Jorge, just to show a couple more slides? I can finish with this as well.
Yeah, I think you're okay. Let's do that.
Yeah, perfect. So I just want to give you a glimpse of some results that we got in the last couple of months, where we say, okay, can we generalize this even further? Because in principle, this can be valid for any system like basketball, right? Where the function of the collective is delivered by a system of interacting components. And we found this data set. Well, this is work performed by master student Flores, who was supervised by Minke. She's a PhD student in my group. And here, our structure function landscape was to map combinations of antibiotics, so antibiotic cocktails to killing Staph aureus. We found this data set where they actually measured different combinations of 6 antibiotics. Long story short, this is what we get. This is, again, global epistasis patterns. You can see that for each antibiotic, there's actually 2 fits. This depends on whether the antibiotics are cidal or non-cidal. And this is how the predictions look like. With this, I want to thank you all for your attention, and I will be happy to take any questions. Just to say thanks to all collaborators and the members of my group currently. And with a nice picture of Delft, which is a very nice city where we are.
Thank you, George. It was really nice to see quite a different approach to predict microbial community behavior, different than the one we use normally in the water or the wastewater field. We have, I can have a short one because we want to keep it in time. I wanted to ask you, because when you quantify this fitness, you see this linearity, but the way you quantify and the way you define this fitness, can it impact the way the function looks like? Because you're looking at concentrations maybe or productivities. Is that in a batch system? Is that in a continuous system? I was thinking a bit of that, how that could imply what could be the implications in this linearity that you show, if you define this fitness in a different way?
In principle, this is a purely empirical thing. So one thing I want to stress is that there's no like theoretical reason or fundamental reason why these patterns must be there. You can build, you know, landscapes in the computer, simulate landscapes where there's no such correlations, or where you just get flat lines. Or, but of course, then how you measure the function is gonna impact the patterns you get. And for different functions, you're gonna get different patterns. Probably, yeah. Or if the functions are very correlated, then you're going to get similar patterns. But the main thing here is to compare apples with apples, right? So if you measure, you need to measure exactly the same function for all your experimental points, right? But in principle, that function can be anything, right? Anything you can quantify. I don't know if that answers.
Yeah, I think that's good. Regarding the QMP, if we put it in the context of a wastewater treatment system in which conditions are changing or could be changing, so how could you see this sort of incorporated into these models? Maybe with a variable P and a variable Q, or we could connect some mechanisms to these probabilities somehow. What's your take on this?
So obviously, Q over P is on average 1 in a wastewater treatment plant. But it's varying from day to day as the conditions change. But what's really bizarre is that nothing actually goes extinct, and nothing seems to successfully invade either. Things are in actually perfect balance. And I would go into that more. And I think there's some big questions to be asked about how such a perfect balance is maintained over time. But because you're right, from day to day, the microorganisms go up much faster than you would expect if you actually had a Q over P of 1. That's just a long-term average. Day to day, it's all over the place. And I could show you data showing how the growth rates and death rates vary over time, but around a value of 1. So but I think the way we would use it in the short term is by understanding that if you want to invade something into a system, then you have to get in there and change the Q over P, either by changing the environment or by changing the organism. It's probably easier to change the environment. But yeah, and there may be ways to predict that.
There's a question about, I mean, you presented the approach in systems which had a known, let's say, metabolic network, or with one or two organisms in there. What do you think the limitations will be to this approach when we are dealing with more complex microbial communities like in sewage treatment plant? Do you think there is a way into here? It's going to be difficult? What's your take on that?
Yeah, that's a great question. So yeah, I think genome scale metabolic models are really powerful because they can capture a lot of nuances of microbial metabolism that empirical models like Monod miss. But they get the parameter space is so large when you start to get thousands of species. And so I think that's where trait-based ecological modeling is a nice hybrid, and you could actually combine trait-based with genome scale metabolic modeling. And so with trait-based modeling, you'd want to group organisms that have some shared physiological trait. And so you might be, we have to simplify at some point. And so doing that, you might have a genome scale metabolic model that represents a trait-based group that maybe has a higher yield or higher growth rate or something like that. And so it depends on how you define traits, but that is one way to reduce parameter space, is to start to group organisms that you think have a shared life history strategy.
So let's, I think we can move on with all speakers. If you would like to, guys, reconnect again with your microphones and cameras and everything. The idea of this webinar was to bring in new approaches or different approaches of modeling. We hopefully didn't have the opposite effect of scaring more the people, because I know that some researchers are scared of the idea of modeling because it's kind of a hard topic to get into. After listening to these presentations, hopefully we did not scare, but interest many of them that there is a field of work that in which there are new approaches still to be explored and they have potential. I think we can all agree that they are not, I mean, many of the questions are regarding the application, how this can be applied, how this can be directly brought into wastewater treatment plant. Perhaps those 4 approaches that you guys presented are not there yet into the application. At the moment, the useful models we have, Rebecca presented it very well at the beginning. There are models that are going to be useful. And up to date, we use the conventional ASM and ADM models, and they are biokinetic models, but they have their limitations. So how can we bring in something new? I think you guys put it together 4 avenues for this. So I wanted to take on some of those questions. Perhaps, how do you guys think? There's several questions regarding how this could be applied to predict performance of sewage treatment plants, which is the golden question in the MIWA group. I don't know if someone wants to say something about this. What's the path for application of this? Tom, you've been doing this for longer. Maybe you want to say something.
I think that I do work closely with the industry in my country, and and they are just about adopting concepts this community developed 20 years ago. I don't think we necessarily have to worry or even worry if what we're doing right now isn't immediately applicable. And I think a thorough understanding of these fundamentals is actually really important to giving a firm bedrock. Having said that, the technologies we have at present are pretty old. Not as old as I am, obviously, but 100 years old, or more. And I think one of the big advantages of the technology is not going to be necessarily in making what we work with right now work better, though they will help with that. But it will really help in actually generating the next generation of technologies that do things we can't currently do with the existing wastewater treatment technologies, and be able to develop those a lot faster. So at present, we're very reliant on developing technologies essentially through trial and error. And I think the methods we have now will allow us to develop new technologies a great deal more quickly. There's a very good essay by a guy called William John McCorn Rankin, and you may have heard of the Rankin cycle. It was him, and he wrote a very good essay on the use of theory and models in engineering, and he highlighted obviously the way in which they can help you take decisions, but also the way in which they can stop you doing things which are stupid. So I think we definitely have that advantage. So yeah, for me, the big advantage will be in developing new technologies more quickly, and in maybe preventing people from doing things which are simply not going to work. Those are my 2 things. Lots of the stuff which has been long known by this community is still not taken up in the world of practice, and we need to work harder at that. So literally the work we were doing 20 years ago. So yeah, that'd be my response.
Yeah, I mean, and I was going through the different points you guys presented, the different approaches, and I think the goal of what we're making the models for is going to be bringing one or the other up to a higher focal point. If we're trying to understand the function, if we're trying to understand the community, if we're trying to understand from the probability point of view that you were showing, I think each one of those could be more central. and depends again on the objective. What will be more useful depends on the question we are asking, and in the context of sewage treatment or other systems. Do you guys want to add something else? Someone else about this application path?
I do want to add something regarding this, because I was thinking that, especially when I talk with experimentalists or people that in their daily life they don't use models, I see they don't see models as a tool that they can use for experimental design. And I think this connects with what Tom was saying right now. Maybe our work as a modelers is also to show that the tools that we create, they can be used eventually for experimental design. It's much faster to run a model than to run an experiment, and it can save us time and money, right? So I think we have to work towards putting our mathematical descriptions at the level that allow us to at least make a screening of conditions, communities that we can play with for a specific objective in the lab.
If I can just add to that, I think that I totally agree with that, and what the models provide us is a theory, right? And theory provides us with expectations. So that's extremely important in order to, well, as Rebecca was saying, to design our experiments, to know where we're going, to know what we're expecting, right? And whether to be surprised by the result we're getting. And in the end, to advance also the application.
I also want to add that we're sort of in the water and wastewater industry, we're getting into the era of process intensification, right? So land is more expensive, treatment processes have to be smaller. We're essentially asking more out of our engineered communities. We need them to do more with less. And so in the past, these systems have sort of been over-designed, excess capacity. They've just been sort of cruising along and maybe not pushed to their limits. But now we're essentially pushing these microbiomes to their limits, and that really creates a need for advanced modeling, because now we're interested in maybe not just making methane, we're in making other products and shifting metabolism, right? Or we're interested in modeling greenhouse gas emissions like N2O. So these are things that are mechanistically, or they have a mechanism, but in sort of maybe more empirical models like Monod are difficult to capture and predict. And so that's where I think we're a little bit at this turning point where we need to, we need some tools to help us actually guide to how do we predict what microbiomes are going to do when we start to push them to their limits.
Thanks. So I think we're gonna have to wrap it up. I hope that at least the message to the audience is that with modeling we can do a bit better than trial and error. And I think I would say that we don't do modeling because it's easy. We do it because it's hard. And I think we do it also because it's fun. So hopefully with these new ideas, more people will be interested in jumping into this field. Pick the way to do it that you prefer, and hopefully you enjoyed the seminar and we hear from you at some point. Thank you very much to everyone. Thanks to the panelists and thanks to the audience. I will meet you hopefully in the upcoming conferences. Just have a great week. Thank you very much.